Thank you very much, everyone, for the break. I hope you enjoyed the coffee, the pastries, and at home, maybe you had them. I don't know, but up to you. So our next speaker is going to be Guillaume Lajoie. He is an assistant professor in the Department of Mathematics and Statistics at the University of Montreal and a core academic member at Mila. He is also a SciPar AI chair and Canada Research Chair in Neural Computation and Interfacing. He will be talking to us about ML for neurotechnologies. And his research group, more generally, works at the intersection of AI and neuroscience, or neuro AI, developing tools to better understand neural networks, as well as algorithms for brain-machine interfaces, or scientific and clinical use. Today, as I said, he'll be sharing some research insights on ML and neuroscience, or neurotechnology, I should say. Guillaume, the floor is yours.
Thank you very much for the great introduction. I have actually one correction to make, because starting from today, I'm now an associate professor. I got tenure today. So this is a
congratulations.
Very happy about that. Now, okay. So thank you for the introduction. Thank you for coming. This is always a great pleasure to have like the broader Mila family come together and talk and share about these research endeavors. As was mentioned, let me see here. This should be working. Here we go. My broader research interests lie at the intersection of neuroscience and AI, right? And so fundamentally, I think there's very important questions that are common to both. Questions about computation, questions about intelligence. And I really think that this multidisciplinary interaction between these disciplines yields better understanding in both, right? And that's not just, getting inspiration from the brain to build better systems and using tools from AI to understand the brain. These things are fruitful, that's for sure, but there's really core common principles. And that's what my lab works on. And so a big part of my research is in theory and in learning theory in general. Part of that program is centered on applications, specifically applications towards neuroscience. And that's what I want to talk about today. So, if you're interested in discussing theory and a lot of these scientific discoveries, I'm happy to talk about this. Today, I want to focus on a more applied version of this sort of intersection, and that is about neurotechnologies, right? So neurotechnology is kind of on the rise. Like if you just look at the news in this past week, for example, you might have seen two big headlines, right? So one of which is, brain implant helps paralyzed man to walk again. It's pretty cool. Another one is, as always, Elon Musk's sort of outfits making the news. So Neuralink gets FDA approval. This is just, signals that there's some progress, some fast progress in developing ways to directly interact with the nervous system. And although this is, seems like science fiction, and it kind of brings us closer to these types of ideas, I think there's tremendous opportunities beyond the obvious here. Beyond just repairing motor control, so on and so forth. And I'll touch on this a little bit more, how neurotechnologies can replace a lot of the treatments in clinical settings and make clinical interventions more precise. So just usually, what do I mean by neural technologies or neural interfaces in general? Let's take the example of this news with this brain implant that made the news, because it has a lot of these components. So usually it's technology that interacts in a closed loop with the nervous system. In this particular case, what the researchers from EPFL did is they implanted electrodes in the motor cortex of a paralyzed patient. They could read signals from the motor cortex of that patient, and then they use it to stimulate the spinal cord below the sort of break. So that's the patient that had an accident, lost the use of his legs, and then essentially building a digital bridge to control to help stimulate the spinal cord and then recover the use of legs. So the components here are kind of twofold. You need to read out information from the brain. So you need hardware to do this. I'll talk about this in a little bit. You need to be able to decode these signals in a way that can translate this in volitional control of a stimulation, then that's the third component, into a control problem. Essentially, how do you reconnect with the spinal cord, right? So there's a lot of moving parts here, and you can imagine that a lot of these things wouldn't be possible without machine learning algorithms to help that pipeline. So I want to talk about a little bit of these progresses today and how we're involved in this field in general. And I want to note here that this general idea of directly interacting with the nervous system is not only for sort of motor diseases, right? Oftentimes, the nervous system regulates a lot of things in our bodies, and intervening at the nervous system can replace pharmaceutical interventions, right? Pharmaceutical interventions usually are kind of a big hammer, right? You take a pill, it goes everywhere in your body, even though you want to target a small part that is problematic. And so if you can target more, interventions that are more local, then you avoid a lot of side effects. So there's hope to use neural interfacing for a bunch of different ailments that are not just neurologically related, in a sense, right? And I'll touch on this in a little bit. Okay, so some disclaimer. This talk will cover some sensitive experimental content, right? So we're talking about experiments that involves the nervous system. So I'll show some photos. Maybe if some of you are queasy, I'll wave my hands and you can cover your eyes for the slides that these are coming in. But things to note, there's no pain receptors in the brain, okay? And the brain is surprisingly robust and resilient. So when we talk about implants, know that there's very well-established protocols and that there's usually no pain associated with it, right? Okay, that being said, let's start from the start. So Danilo did a great job at motivating the neuroscience and the importance of neuroscience in the modern scientific field. And so let's take a step back really, and so we're on the same page. In the brain, information is processed largely by neurons. Neurons are cells that communicate with each other through synapses, right? So they're units that are connected with each other, and the synapses act as weights, right? And these interactions are mediated with electrochemical activity, like spikes, action potentials. And these form big networks akin to, obviously, artificial neural networks, and they process information. One thing to note is that evolution learned basically general recipes to wire neurons together and for them to learn functions, right? And as Danilo mentioned, some of the parts of the brain that are sort of higher up in the evolutionary hierarchy of humans are dedicated to learning, right? To like basically they're kind of useless at birth and then they learn a lot throughout development. And the same is true to a different extent in the sensory systems of many animals. And I want to make the point that the general recipe is pretty much the same across a lot of animals. And so that means that whether you're an electric fish or you're a rabbit, a lot of the sensory inputs you get from the world are integrated sort of like in the same way, right? There's neurons that integrate these signals. And so, and you can repurpose these things. And there's evidence of this in nature, right? Blind people repurpose brains, visual areas for language. There's plasticity here that is the general recipe from evolution. And so that tells us that in principle, we should be able to write information to the brain and to read information from the brain pretty easily, in the sense that evolution has granted these solutions that facilitate this. So as long as you have a clean signal, the brain should be able to learn to interact with external devices, right? And so this is this paradigm that I want to explore today. Okay, so brain-computer interfacing, what does that eat in winter, like we say in French, right? So definition is essentially directly interact with the neural activity in a closed loop paradigms. But why would we want to do this? Well, there's a few different reasons. First, as I kind of alluded to, there's medical applications, right? The development of prosthetics, to mitigate pathological neural dynamics, promote recovery, all topics I'll cover in examples today. The second, and I think that's a really important one, is that it's a magnificent scientific tool. It enables to probe and to interact with the nervous system to understand how brain circuits work, and helps us ask scientific questions that go beyond usual experimental paradigms. And I'll quickly touch on this at the very end if I have time. And the third, of course, is it changes how we interact with technology. We can control devices in ways that we couldn't before, and so on and so forth, right? Okay, so how is this done, right? So when we say brain-computer interfacing, there's a big spectrum, right? And so I'd like to kind of put this on a map with two axes, like we do usually, right, to make things overly simple. And so on the vertical axis here, I'd like to point out that there's basically two modalities. One is invasive, i.e. something that you need to get implanted, goes under your skin somehow. And the other end of that axis is non-invasive, a helmet you put on, like a bracelet, right? These are different ways to interact with the nervous system. And on the horizontal axis is kind of the way information flows. You can either input information in the brain, stimulate the nervous system, or read out information from the brain, decode from the nervous system. And of course, like in the examples I showed at the beginning, these two modalities are kind of used, right? Like you're recording and then stimulating in a bidirectional way. So there's like a host of different modalities along these axes, right? So on the input side, you can think about, in non-invasive, you can think about ways to stimulate the brain that don't require implants, like a transcranial direct current stimulation and TMS, which does this with magnetic fields. And of course, like all of the technology we use, right? Your phone, your computer is a non-invasive way to input information to the brain. You're using sensory inputs, right? And then on the output sides, there's all kinds of technologies to read out information in the brain that's non-invasive. EEG, cortical hemodynamics, EMG, which are electrodes that read out activity from muscles. And of course, there's the big rigs like fMRIs and so on, right? And now on the invasive part of things, this is where it gets a little bit gory, but I think the important part to understand is that this is how to get the real, like true clean signals, right? There's a lot of noise in the non-invasive part of things. In the invasive part of things, the price to pay, of course, is to go through surgery, but you get really a lot more degrees of freedom to play with. So on the input side, there's optogenetics technology, which is essentially uses light to activate neural activity, microelectrode stimulation, and ECOG, right, which is a form of surface electrode that's on the surface of the brain, which is the modality that was used in the first example I showed. So this is kind of the landscape. So it's very multimodal and it's very active landscape for different use cases. You might want to use different modalities, and I'll touch on challenges that kind of address all of these modalities. But before going too deep, I want to talk about early history of brain-machine interface. And one could argue that one of the first time a brain-computer interface or brain-machine interface was created is in the 60s, actually, in this seminal paper from Eberhard Fetz, where he demonstrated through an experiment that is called operant conditioning, that essentially it's possible to control the activity of single neurons in your brain if you have feedback about that activity, right? This is a pretty cool experiment. So what happened is there was a monkey that was implanted with an electrode in its motor cortex or sensory cortex, I think. And he was given, like, he was able to look at a dial that was giving it the firing rate of that neuron, right? And essentially the task was, if you match a target firing rate, you get a reward. Very quickly, the monkey learned to control the activity of that one neuron that was recorded from the brain to get that reward. So think about this for a second. That means that just by having feedback of what the firing rate of that single neuron is, you can learn to control that in a very volitional way. And this is reproducible across the board, right? Again, it points to the generality of the sensory motor type of cortical dynamics that we are all endowed with, right? These things, if you learn a task, you practice it, you get error signals, you suck at it for a while, and then you get it. You play tennis, like you get to hit the ball. Same is true if you plug in a different modality, right? So this was the first time this was shown, as this was in '69, right? So we've known this for a while. Fast forward now, there's a lot of neurotechnologies that are out there. Deep brain stimulation is a common treatment for Parkinson's disease. Cochlear implants are another type of implants that are quite used in treating sort of hearing disorders. These are always things that interact directly with the nervous system. And from the industry point of view, there's a rapid development of hardware. There's a lot of research in academia, a bunch of companies that are developing this hardware to make interacting with the brain and the nervous system more direct, right? So just an example, things are scaling up quite fast. The chip from Neuralink here has sort of 1,000+ recording channels, which is a far cry from that single electrode that was used in the '69 experiment. So this is an example. You might have seen this a couple of years ago, which is the sort of demonstration from Neuralink that shows a monkey playing Pong with the neural implant. Again, the idea is that the little rod that the monkey is holding here is just a reward. But the activity of the paddle in this Pong game is controlled with a neural prosthetic that's implanted in the motor cortex of the monkey, right? So you might have remembered this. So this little part here is controlled by the monkey that's learned to control it and learned to play Pong. It's quite astonishing. But when you think about it, this is actually the same thing as what was done in the '60s, right? It's 1 degree of freedom control of an external device, right? And so this was sort of the state-of-the-art demonstration of what we can do with these implants. But really, there's not a lot that's changed into what's been demonstrated before, which brings me to my point, right? Our decoding and encoding capacities does not immediately scale with channel counts. There's a huge challenge into leveraging these extra degrees of freedom when we're talking about interacting with the brain, right? And so you might have seen other types of results like this, where a patient was implanted with a neural interface in motor cortex to control an external device. These types of early demonstrations heavily relied on primitives that were learned on the robotic side, right? So it's very light interactions from the nervous system. In other words, it's not like that lady is controlling all of the joints in that arm. This arm was optimized to do so. And it's arguable that we want to go beyond this. We want to involve the brain in learning to control high degrees of freedom. And there's a big question of how to do this. We have the hardware that's coming online, and we need to develop machine learning strategies to take advantage of this, right? And so here are quotes here. A lot of people agree, and these are old slides. Maybe I should remove the Musk quotes here. Challenge to design adaptive algorithms to maintain reliable and robust performance while continuing to improve over time is a major challenge. And another quote from Thomas Reardon, who's a co-founder of Control Labs, which has been acquired by Meta, is the mother of all machine learning problems. Okay, and so why is this, right? So I want to identify a few challenges for ML to be applied to neural interfacing. First is that neural signals are variable and noisy. Like it's biology, it's messy. 2, every brain is different. It's not like you can train a huge deep network on the average population, stick it in a new person and expect to have like magic learning transfer learning, I should say. Third is that neural circuits are not stationary. They actually learn. That's why this whole thing works, right? So you're dealing with a problem that you're trying to interact, you're trying to learn a two-learner sort of problem, right? As you learn to interact with something, it also learns to interact with you. And so that's a challenge as well from the machine learning point of view. And fourth, and importantly, there's very limited data. It's expensive to do a kind of like systems identification sweep on neural interfaces. You can't just probe all the parameters that you can, right? Like this is an expensive thing to overcome. So this is all that to say that on the face of it, it's not necessarily directly well suited for deep learning approaches, right? So we need to go back to other types of machine learning approaches that address these challenges. Okay, so what I want to touch on today is a few examples of work in that sphere from my lab. Happy to talk about, take questions on these as we go along, right? The first of which is optimization of intracortical stimulation for motor function recovery. Okay, so this is a collaboration with Marco Bonizato and Numa Dankos from the Polytechnique and the Université de Montréal, who both run experimental labs in this domain. So to motivate this, I want to say that each year there's about 50 million people that suffer from debilitating motor system injuries. So that's stuff like spinal cord, stroke, that leads to loss of motor function, right? And it turns out that targeted stimulation of motor pathways can actually speed up recovery, right? By targeting circuits that have not been damaged but are weakly connected to what you want to do, you can actually kind of recover use of your hands, of your legs. And that's essentially what physiotherapy has been doing for years by forcing the movement itself. But it turns out that if you target the nervous system in a very precise way, you can speed that up tremendously. Okay, so this is a control problem. The idea here is you have an implant that interacts with the nervous system. You have the ability to stimulate the nervous system with a family of inputs, and you want to get targeted outputs. For here in this example is motor related, right? So here you have at your disposal a family of parameters that you can decide where to stimulate, the amplitude, like what type of timing your system, your signals are taking, and you want to get to desired target activations. And so the challenge here is that the parameter spaces are large. If you have large degrees of freedom in your stimulation paradigm, then the search space is big. And you have limited queries. You want to do this fast, right? And so the objective here, what we've been employing is an objective-based optimization where we want to target some sort of optimal activation or observable. Okay, so we've been using as a first pass, the good old tried and tested Gaussian processes, which have been quite useful. So just a quick point here about Gaussian processes. It's sort of a nonlinear regression type of schemes that actually gives you an estimate of uncertainty, which is quite useful when you're doing some space search, right? And so I know Gaussian processes have been heavily developed for geosciences as well. So the previous talk that has addressed using them in some sense as well. And so the experimental design I want to describe right now is basically that, trying to find a way to rapidly identify stimulation patterns to evoke specific muscle activations, right? So here you want to stimulate in the motor cortex of a subject, and we're recording the muscle activity of in the wrist of the subject, and we want to target activation of specific muscles at specific timing. So the way we do this is we've developed a kind of a novel hierarchical Bayesian approach that combines priors of Gaussian processes in a sort of sequential way to speed up this exploration. Okay, and so, for example, if I'm recording activity from two muscles, you can see here muscle recorded from EMG0, muscle recorded from EMG4. And we want to have activations separated by some time tau, right, as kind of a complex movement. Then we would like to, search the space of stimulation of an array that's been implanted in motor cortex to evoke these muscle activities. So if we use standard Gaussian process, typically this is the number of query points we need to get to a mean accuracy of about 50, right? But with our hierarchical approach, essentially what we do is we first fit lower dimensional Gaussian processes and then leverage the kernels that have been learned through Bayesian optimization to combine with higher order priors. And we do this iteratively. It turns out that you can speed up and actually get way better convergence on these optimization schemes, okay? So, and at the end, this is the true data. So you see we've evoked activity in muscle, the blue muscle here and the orange muscle here in ways that are differentiated. Like it's not like we evoke all muscles at the same time, and really close to the target that we've been aiming for here. Okay, so again, the idea here is that you build hierarchical kernels and you use them as priors as you build your Gaussian processes for the full thing. Okay. So this has been published as an algorithm in this paper here. And just recently, we've applied this algorithm to address optimization for natural movement. So this is a paper that appeared in Cell Reports Medicine last month, actually. That's the result of a long experimental journey where we showed that this algorithm can work online with natural movement, both in flexion extensions of a hand and also in a gait in a walking situation, right? And so I'd like to show you a little video here that shows this algorithm in working online in real time, right? So here, what you'll see is that there's a rat on a treadmill that has a spinal cord injury. So you'll see that the spinal cord injury is partial and the leg is dragging as the rat moves, right? And now the rat has an implant here in motor cortex, and in real time, we're tracking the foot elevation here. So now here, the previous example I showed you is we're tracking the activation of the muscles themselves. Now the observable we want to optimize for is something that's more complex. It's actually the level of the foot, right? And as we go along, what you'll be visualizing here in the bottom right is that there's a search of the parameter space that's done through this Gaussian process hierarchical algorithm. And essentially, this agent learns new combinations of stimulation through that array that leads to recovering of that gait, right? So you'll see here, we're tracking the foot elevation. We're selecting the best stimulation strategy that's timed with the gait of the animal itself. And then you can see that you've restored, you can see the difference between before and after here. Okay. So here, what's interesting here is that this is an injury in the spinal cord. It's partial, so there's still circuits that send signals to the muscles, but they're sort of unused, right? So we're recruiting new circuits in the brain that are weak, but by stimulating them, you can evoke you can recover walking procedure. And it turns out that if you do this long enough, then you're promoting these circuits to recover healthily too, right? So it's not like you need this to be on all the time. It's a tool for recovery. Okay. So in summary, right, for this part, right, I've shown you that Bayesian optimization with Gaussian processes offer efficient online stimulation search for fixed objectives. We can reveal circuit computations, and it's easily scalable to more complex spaces. Okay. And I should say actually that this led to actually a Mila startup called Neural Drive. So Marco is the CEO of that startup, and so we're working towards bringing this to a clinical setting. Okay. How am I doing on time here? I'm good? Okay, cool, perfect. Okay, so I want to switch gears here and talk about treating disorders by modulating the peripheral nervous system here. Okay, so this is, speaking of startups, this is a collaboration with Mila partner startups here, BIOS, with whom we have ongoing collaborations. And thanks to MyTax and MedTech, which are granting programs here in Quebec, we were able to fund these collaborations, and we're really happy how things are going. And there's also some research that's done in collaboration with the Feinstein Institute here in this part. Okay, so here again, switching gears, the idea is that, as I mentioned earlier, there's a lot of things that happen in your body that are, modulated by the nervous system, right? Cardiac rhythm, your breathing, a lot of metabolic function are mediated by the nervous system. And so a lot of pathologies that are linked to these functions can be addressed with neural interfacing, as opposed or in combination with pharmaceutical treatments. So one of the most popular ways to target this peripheral nervous system is by interfacing with the vagus nerve. So the vagus nerve is kind of the highway that goes from your brain all the way down and innervates a lot of your organs, your heart, your lungs, your liver. And so this is a part of the parasympathetic system that essentially links your brain to a lot of your organs. It turns out that these stimulating the vagus nerve can have tremendous impact on a bunch of different diseases, and it's been in use experimentally for quite a while now. And basically, it involves very minor surgical implant of cuff electrodes along the vagus nerve that enable to record and stimulate the nerve. And so the challenge here is that you want to optimize different ways to interact with the nervous system, with the vagus nerve in this particular case, which is kind of a bundle of a bunch of different nerves. So it's kind of a deplexing type of problem. You want to be able to record activity from different fiber bundles that go to different organs. And you want to do this to not necessarily have sort of an online prosthetic that just stimulates all the time, but the first step is really to give recommendation tools to surgeons, to clinicians, to better tune these implants for targeted use. Okay, so the way we do this is again, in the same way we use Gaussian processes with Bayesian optimization to really optimize these interventions, targeting different biomarkers, heart rate, breathing, blah, blah, blah. And we do this pretty efficiently using Bayesian optimization. These are plots here showing that Bayesian optimization of these things actually do a much better job than doing some random search and some other kind of baseline stimulation tuning searches, basically. So what I'm showing here is a map in, for example, current and width of pulse stimulating this the vagus nerve, and we're measuring the activity in specific fiber bundles that are linked to specific biomarkers here, right? And on the left, we show the activity that can be recorded from different electrodes down the line. You can see that's kind of a multiplexed signal. There's different fibers that send information faster and at different depth. And so part of the challenge here is to decode which features of which of these activity patterns that we're able to record are linked to different biomarkers, right? So all in all, there's a big pipeline that's being developed to predict when certain pathologies are going to arrive from reading from the nervous system and how to intervene to optimally treat them, right? Within this pipeline, right, and so there are kind of other challenges, right? So if you have this pipeline, you're fitting some sort of stimulation parameter to an outcome. You might want to do this with online optimization in a new patient. That's great. We have ways to accelerate this. But in a sense, you're still starting from scratch every time you have a new patient. We know broadly that these things are similar across patients, but there's an opportunity here as you get cohorts of more and more patients that are implanted with these treatments to leverage this knowledge, right? So it's not exactly possible to do rapid transfer learning. But the question we've been asking here together with Stavros Zanos here, who runs a lab at the Feinstein Institute, and my PhD student, Chimeng Mao, is how can we accelerate personalization of stimulation protocols, given the fact that you might have a cohort of past patients that have been implanted, right? So what's important to understand is that there's actually a lot of variability across populations. So what we're showing here is experimental results from the Zanos lab that shows relationship between different biomarkers. Here, EMG amplitude, this is recording from a muscle, percentages of heart rate impact, and this is breathing data, so delta and breathing rhythms. And then we're fitting this to fiber amplitude, right? So if you're recording from a fiber in vagus nerve, you have a different impact on these different biomarkers. And then this is data in rats, and you can see that different colors have quite different fits, right? Generally speaking, you can see that here linear fits are better, here exponential fits are better. But overall, there's a lot of variability across subjects, right? So how can we navigate this in an efficient way? So what we want is, again, leverage this variability and similarities across the population to rapidly fine-tune an optimization strategy for a new patient. So you might want to start from a pool of population. That's a weird term. We need to change this. Pool of subjects. And then you might want to derive a population fit. What's the average fit that you want that fits the general population? And then you want to rapidly adapt this for a new individual in order to jumpstart your optimization process. And one way to do this is actually to leverage meta-learning ideas, right? Meta-learning I'm sure a lot of you are familiar with this, but here's a hasty overview, right? Meta-learning is basically a way of learning to learn. So if you have a system and you want to learn multiple tasks, for example, you might want to initialize a system in ways that it's easy to learn new tasks, right? So you might have a set of what we call meta parameters. This can be initialization, it could be optimization settings, in a way that multiple different tasks down the line can be learned faster. So the meta learning cycle here is choose these parameters, then optimize on different tasks, right? Task one here, this meta learning example is learning to dissociate different things in images. For example, dogs from music instrument in task one, and barrels from cliffs in task two, and so on and so forth. So the question is, given these meta parameters, how can you rapidly tune the parameters in specific tasks in your inner loop to learn faster, in a sense? And so there's been a lot of research in meta learning for multitask learning and in AI in recent years. And the idea, again, is you basically learn in hierarchies, right? You want to optimize parameters that are good to optimize parameters. And it turns out that we can use these paradigms if you think about learning multiple tasks as learning multiple fits for different subjects, right? So what we've developed here is a way to essentially learn a meta parameter set, which is the set that's associated with the population, that best is rapidly learning a fit for a new subject, right? So this is the pipeline. I don't want to get too much into detail, but we initialize a model with this meta-learned population fit parameter, and then we can rapidly, in the inner loop, use Bayesian optimization to optimize for a specific subject. And it turns out that you can fit even with very few shots. That means if you query your space of parameter with just 2 or 3 points, you can estimate a fit much faster than with traditional method if you didn't use the population. And even if you use the population, but with naive sort of transfer learning methods. So this is very promising. And we've done it in vagus nerve simulation, but this is an approach that's useful across many ranges of neuromodulation technologies. Okay, so we're really excited about this. These are results. Our different methods here, meta-learning GP and meta-learning regression do better. I'm going to skip this. You can trust me on this. And the summary here is that peripheral neurostimulation can be optimized via Bayesian optimization, which is a good paradigm to use, in my opinion. And meta-learning is flexible framework for neurostim personalization, and we can be included in online optimization protocols across a wide range of use cases. So we have a paper that's currently under review on this. Okay, lastly, I know I have like about 4 minutes. I'd like to talk briefly about another collaboration that I have. And this time it's more geared towards scientific discovery. And that is surrounding the topic of identifying and guiding learning dynamics in the brain using brain-machine interfaces, right? And this is in collaboration with Dr. Amy Orsborn, who runs a primate lab at the University of Washington in Seattle, and my postdoc, Alex Payard here. And there's a few other people in my group working on this collaboration. And this is more of a readout task. So the two first examples I've spoken about is about, stimulation, inputting information in the brain. This is decoding information to the brain to control external devices. So here's the experiment. That's quite a standard experiment in motor neuroscience. The task is that there's a screen presented to, in this case, a monkey, where there's a target in the middle and there's 8 peripheral targets that light up randomly at different trials. And the task is to move a cursor on the screen from the center to one of the targets to get a reward. It turns out that you can teach that task manually with and then with the help of cortical motor implants, the patients can learn to control the cursor on the screen pretty rapidly. Again, this is a 2-degree freedom task. It's quite standard now. If you read out from enough neurons in motor cortex, you can learn to control a cursor on the screen pretty easily. Okay. Now the question is, can we use these types of paradigms to really probe how learning happens in the brain? Learning happens in the brain. We have a pretty good idea of how it does at the level of synapses. We have good understanding of synaptic plasticity, but how this turns into learning in neural population is not quite well understood. And these BMI, again, the question is, what are the computational principles of learning in neural population at large that coordinate these plasticity mechanisms? And our solution here, our approach here is to use brain-computer interfaces, which actually give an unprecedented access to this learning paradigm. And by that, I mean, in any neuroscience experiments, if you're recording from the brain while an animal is learning to do a task, what you have is at best correlates. Right? You're correlating the activity of your recorded motor neurons with, a reach or some sort of task paradigm. But there's so many neurons you're not recording from. There's a lot of stuff you don't know. You can't record from all the neurons. In a BMI task or BCI task, you have explicit access to the readout. What controls the stuff outside is only the stuff you're recording from, right? Because you're using that information to drive a cursor on the screen. There's nothing else. There's no spinal cord that leads to muscles to whatever, right? Like you're directly recording exclusively from the neurons that are outputting to the world. And so you can ask questions, directed questions about learning in much better ways. You can perturb outputs of readouts and see how the brain adapts and so on and so forth. And this is the suite of experiments that we're developing here. And specifically, we're interested in the idea of co-adaptation. If there's learning that happens on the decoder side, say from a neural network, how are the representations that are learned by the brain to adapt to this learning going to evolve? Can we target certain types of representation, higher dimensional, more robust, by doing some interventions on the output side, such as regularization, using tricks for machine learning? These are all questions that are actually quite fascinating. Basically, how does regularization and tricks from machine learning impact learning from the brain? And we can address them in this paradigm. So the idea is to couple these the ideas from neural network theory with learning paradigms in neuroscience to probe really how the brain works and design these neuroplasticities to scale better, right? If you want to use these to control more degrees of freedom, you need to be able to understand how learning happens in the brain. That's the key. Okay, and I'm giving the signal that I'm close to the end here. So these are kind of the big questions we're looking after. What computations drive learning in populations? We use deep networks to probe this, and how sensory feedback inputs in multiple areas contribute to learning. So how do we integrate error through our senses? These are computation theory efforts, different programs that are happening in my group surrounding that project. We're trying to infer dynamics, learning dynamics from neural activity. We have neural theory of manifold and gradient learning based on data and theory that Alex has been developing. We have a preprint out there. And then we're also developing plasticity models to compare different bioplausible learning paradigms with what we can observe in the brain. Okay, so this is my summary. I hope I've convinced you that BCI technologies at scale are coming. They have substantial potential for impact, both in clinical applications, provide unique scientific tools, and also new ways to interact with technology and AI. And flexible AI ML approaches are needed for these unique properties. Namely, we have unique type of variability, non-stationarity, stuff learns, and co-adaptive paradigms between AI and the brain are kind of the key to many of these things, right? And I think some of the ingredients of these challenges are shared with many other application fields, right? So if you have an idea about how these types of ingredients can help you in one of your problems, come and see me. I'm happy to chat. And so with this, thank you. This is all the kind people that given us money to do this. And happy to take questions.
Maybe, I don't know if there's any questions online, and please feel free to signal, but I have one question maybe on my end. But so you talked about at the beginning that it is coming, well, at the end as well, that it is coming, these types of technologies, but is the adoption of those technologies, how do you see the adoption of technology of this in general public or?
So that's a good question. So there's definitely consumer products, obviously on the non-invasive sides that are getting out there, right? Like we've been hearing about leaks that, like an example, we might know in a couple of days, Apple is pointing towards like AR glasses that have EEG electrodes in them, right? So these types of consumer grade products are coming. There's actually some folks involved in the Mila partnership, I think, like folks at Lucid are using these types of things. So that adoption, I think, is coming fairly fast. On the clinical side of things, there's and for good reason, but lengthy regulatory processes, right? The clinical trials are long. And so things are moving relatively fast on the research side of things, but in terms of deployment for treatment, we have to go through the processes, right? Like the same way pharmaceutical treatments are being developed. So patience is this is really deep tech, right? Like if you're talking about developing these types of technologies, you need to think in terms of 10 years in advance sort of thing. But that doesn't mean that we shouldn't do it, right? Like I think there's a really high reward at the end of that road. So when I say they're coming, it's on the sort of clinical timeline that you need to think. Yeah. And
on it, because you talked about non-invasive on the side of invasive.
Invasive, that's what I mean more. Like non-invasive, it's faster, right? Like you think about, again, like EEG, use of EEG for menu control, whatever. There's also control labs, which is now a branch of Meta that uses peripheral signals like that bracelet I showed earlier. Essentially, it's remarkable how you can learn to interact with devices just by micro movements of your muscles, right? So you can record from the peripheral nervous system, i.e. from your muscles, and learn to be very volitionally control things like a computer and so on and so forth. So these technologies are rapidly becoming available. Yeah.
All right, do we have any questions online, Sophie? No, we don't.
All right.
Thank you very much, Guillaume. Congratulations again on the associate professorship.
Thank you.
We might have a question actually for Guillaume.
I do have a question.
Oh, go ahead.
I've seen in this domain that these peripheral, like the bracelets you were talking about, it was difficult to interface. A few years ago, they were talking about even for a patient, after a few hours or a few minutes, the signal was changing.
Mhm.
So is it still this or what you were
Uh,
talking?
Yeah, I think it's come a long way. And that goes to one of the points I was making, like co-adaptation is key, right? Like both as the patients learn how to use these things and as, the time of day changes, different days, there's drift, there's all kinds of different ways, right? So robustness in the coding algorithms need to be like really on at the top, right? I think there's been tremendous progress in the past few years, right? Most of it has happened, I think, through these big companies. So like transparency is not as great as in the academic circles, but definitely there's a lot of progress in the open source community as well. So that's changed. Yeah.
Okay, thanks.
Thank you very much. Well, thank you, Guillaume. Congratulations again on the associate professorship.